Thanks a lot to the organizers for inviting me. And I hope I can stir your interest in cryptic antigens. Hi. So I would like to introduce to you a novel concept to generate disease-specific antibodies. And with disease-specific, I mean discriminatory antibodies. Antibodies that distinguish a disease-associated form from a physiological form. And such disease-associated protein states can arise as a result of mutations, limited proteolysis, or aggregation. So there is an unmet need for disease-specific targeting. And needless to say, the majority of FDA-approved immunotherapeutics are not disease-specific. So they recognize healthy tissue as they recognize diseased tissue. So the healthy tissue then often, depending on the expression state of the target protein, acts like a sink. And you have to overdose your therapeutic antibody in order to get to increase therapeutic efficacy. To address the unmet need for disease-specific targeting, we have developed and patented a novel antibody discovery platform to produce disease and cancer-specific antibodies. MRCT has transferred complete IP ownership in 2015. And we are basically seeking collaboration with industry and academia to support us in our pursuit to generating novel therapeutic strategies. For our proof of concept studies, which will include 3 cancer targets, EGFR, HER2, and EphA2, we are seeking investment of £1.4 million. So in the next slide, I would like to introduce to you the problem of disease-specific targeting. So my background is in misfolding diseases. And for the Prion protein, there's a whole body of evidence showing that there's a disease-associated form and the physiologic form. That can be analyzed and distinguished in the disease-specific form. We have more beta sheet structure. We have more alpha helical structure in the physiological form. So you would think that's a great model to produce some really nice diagnostic antibodies. And in fact, more than 1,000 antibodies have been produced and characterized in the Prion field. It's your guess how many are disease-specific. What will surprise you is that there's not one discriminatory antibody. Even there's the claim for disease specificity, but that has been rebutted or by others in later publications. So when I started in the Prion field and realized that, I thought that's absolutely amazing. Now that is a complete failure of the obvious. So what are we doing wrong here? So actually Prion diseases is a very interesting model to study that. As I mentioned, the majority of therapeutic antibodies is not disease-specific here, just illustrated by Western blot. Anti-HER1, anti-HER2, they are binding, they are recognizing normal and ovarian cancer tissues. So cryptic or hidden epitopes have been recognized as compelling targets when displayed in disease. And there's a lot of work on cryptic epitopes, and Eli Sarkas published some seminal work to actually correlate exposure of cryptic epitopes and autoimmunity. But so limited proteolysis is an obvious modality to expose a cryptic epitope. And what happens, so basically in the tumor microenvironment, a lot of proteases are active, but they are not chopping up the surface proteins. Sometimes it's a limited proteolysis. What happens with the protein conformation? It changes. So I want to just say two side notes in regards to cryptic epitopes and autoimmunity, because that's really important to understand the bigger picture. So there's a high correlation of the exposure of cryptic antigens and autoimmunity. And Erica Darren and Anthony Rosen have studied in detail granzyme B action. That's a serine protease. And they mentioned granzyme B efficiently cleaves many autoantigens, suggesting that autoantibodies may arise when cryptic determinants are revealed to the immune system. So that means there are T cells in the T cell repertoire that react with cryptic epitopes. So why have they not been deleted during thymic development? That is actually a threshold problem. But then in some cases, if there's a tipping point and more of these cryptic antigens are revealed, the T cells are activated. And that happens in autoimmune diseases, but also in cancer. The second point I want to make is that what is cryptic to the immune system is not necessarily conformational cryptic. As you know, tumor-associated antigens is a whole list of hundreds of tumor-related proteins that trigger a T cell response. And the cause could be that they have been categorized in different groups. It could be mutations, it could be overexpression, or it could be expression in the wrong context. For example, if there's a protein that is expressed during development, then shut down, and it's again expressed in the cancer then it's a problem. So my last point is that traditional antibody selection fails to identify monoclonal antibodies against cryptic epitopes. And I come to that later. A prominent example of a disease-specific antibody is MAB806, developed by the Ludwig Cancer Institute. It's an antibody that has been raised against EGFR variant 3, so that is a deletion mutant. So the approach was clearly hypothesis-driven. Let's produce an antibody against EGFR variant 3, and we have a disease-specific tool. However, the antibody, I don't know, did I mention? Yeah, EGFR variant 3 is exclusively expressed in the brain, in gliomas, not in the peripheral tissue. So this antibody had a couple of unexpected features. And one of the features was that this antibody recognized cancer, colorectal cancer, but not healthy tissues. So they looked at that in detail. So MAB806 recognizes the non-mutated full-length EGFR on a tumor cell, but not on cells from healthy tissues. So how can we explain that? MAB806 is binding to a cryptic epitope on EGFR that is hidden in healthy tissues. The group of Lloyd Ohl did a lot of imaging studies, and basically the cryptic epitope displayed here is displayed in the closed untethered conformation. However, as I mentioned in the title, it was a serendipitous discovery. So it was not a focused approach. So what we are contributing is a focused approach to identify anti-cryptic antibodies. There are other groups who try to identify cryptic sites in silico. So this group is utilizing the protein data bank and MOAT to identify concave patches, binding pockets, and cryptic sites. So the authors claim that cryptosite, which is their algorithm, increases the size of potentially druggable human proteome from 40% to 78% of disease-associated proteins. Not necessarily sure how these numbers come about, but maybe it's a good a tool. So in order for us to conduct or to generate therapeutics, we actually have to target the ectodomain. And one mechanism how the ectodomain can be changed, the conformation I already told you, that is limited proteolysis. But also there's an increasing body of evidence that mutations in the kinase domain, intracellular, feeds through to the conformation of the receptor. And there is new data, quite interesting data coming through that confirms that. So this group has expressed EGFR mutants, deletion mutants, and actually using super resolution microscopy, they show enhanced dimerization and conformational changes of the ectodomain. I'd like to illustrate to you how we identify disease-specific drugs. So under physiological conditions, EGFR undergoes conformational changes, but some changes are maybe specific to cancer cells. We only know of a cryptic antigen or cryptic epitope in EGFR due to MAB 806. So our question was, how do our experiments have to look like so that we can isolate these highly valuable anti-cryptic antibodies? So what we are doing is we are growing cells in 96 or 384 well plates, and we globally change the conformation of all proteins by denaturation. So we have two states, a native state, a denatured state, and we are doing differential testing. So we are then testing antibody libraries, but we are only we're only picking antibodies that do not react with a native state, but that react with a denatured state. We then can go on and test these antibodies that we pulled in vitro and test it in cancer tissue versus normal tissue to verify or validate whether there is a disease-specific change. So with our technology, cryptic antigen display, we can map the conformational landscape of oncoproteins and surface receptors. Importantly, we don't need prior knowledge of any disease-associated states. However, we can also use that in a hypothesis-driven way by expressing activating mutations and then screening antibody libraries. So our technology is disruptive in two respects. We are addressing disease specificity, and we are addressing the failure to isolate such antibodies. Why is that? So in traditional antibody development, the first and foremost action, if you're screening an antibody library, is to separate binders from non-binders. And this is critical because we have so many of the binders. And then the binders will be further validated or characterized regarding specificity, affinity, epitope, function, etc. We at Emerge Biotech are interested in antibodies against cryptic antigens. And these antibodies are, per definition, non-binders. So that actually creates opportunity because we can help pharma to de-risk their therapeutic antibody production. So a cryptic epitope is evident if an antibody does not bind under native conditions, but under denaturing conditions. And also, a conformational epitope is evident if the antibody binds to the native state, but not to the denatured state. The largest number or the largest proportion of antibodies that we have tested is conformation invariant. So these antibodies bind to the native state and to the denatured state. Great research tools. Not so interesting for us. So antigen, so that method on its own would not be valid unless we are interrogating target specificity or monospecificity. Because by denaturation, we bring up a whole new array of epitopes. So we really want to make sure that the antibodies we pull are target specific. Removing the antibody target is the most effective approach to test monospecificity that has been mentioned many times and is plausible. What is not so well known is an alarming publication from Genentech in end of 2012 that was released and where they stated that a considerable number of monoclonal antibodies in therapeutic development show off-target effects. And they measured off-target effects by clearance. So a lot of these therapeutic antibodies in development were cleared much faster than expected. They investigated that in detail and found it's due to off-target effects. So we did a proof of concept study with more than 500 commercial antibodies. And we asked the question, can we identify antibodies, anti-cryptic antibodies? So this anti-actin antibody does not recognize its target under native conditions. So these are nuclei, this is the merge. However, under denaturing conditions, it recognizes its targets and it nicely reveals the actin filaments. The same holds true for an antibody against protein disulfide isomerase. So in this study, we identified 8 antibodies in total that served 2 of our criteria. First, they were monospecific. Secondly, they were anticryptic. An antibody against nucleoporin binds well to the native state. However, the binding is greatly reduced in the denatured state, and that tells us that this antibody is conformational. This epitope is conformational. As mentioned, a large variety of antibodies recognize the native state as well as the denatured state. One question that is often asked is, this denaturation, does that not completely bugger up the cell? And I would like to show you a magnified version of this antitubulin stain, and I chose 2 mitosis to illustrate that. And you see in the denatured state this beautiful tubulin network. So the cell and the intracellular structures are fully preserved under these conditions. So this is how we were going about antibody production. After selecting a cancer target, we were curating antibody libraries, screen them. Our output is antibodies that are disease-specific and monospecific. We can pursue a licensing strategy, or as well further develop the antibodies with pharma MRCT or CAR-T groups. We can also help to de-risk therapeutic antibody development for pharma. So my last slide, who are the players in the lung cancer field? So the Ludwig Institute has outsourced the monoclonal antibody MAB806 to Life Science Pharmaceuticals, and they humanized it, and it's currently in phase 2 clinical trial. Big players in the field of tyrosine kinase inhibitors, AstraZeneca, but many more. Immuno-oncology therapies, Bristol Myers Squibb, Amgen, and more. So what we envisage is that we can use anti-cryptic antibodies or disease-specific antibodies in combination with, for example, tyrosine kinase inhibitors. What are our competitive advantages? We are discovering monoclonal antibodies that are missed by conventional screening methods. The antibodies are disease-specific. That means we can lower the drug dosing, which may significantly reduce side effects. As I mentioned, we can use them in combination with other therapies for personalized medicine, and the platform technology is tailored for discovery of novel cancer targets. Thank you very much for your attention.
Thank you very much. It was an amazing talk. We're running a bit behind, but I think it's such an interesting talk that we have to open for a couple of questions. Yeah, is that it?
So nice talk. So how do you, there are probably many ways you can denature proteins in cells. So some can lead to effect of some kind of completely give a straight, right? They can use some other. So how do you develop those mechanisms?
Well, I mean, that is not our experience. That some can be denatured, the others are completely buggered up. So we have tested. What we are doing is, if an antibody works under denaturing conditions, it basically works on Western blotting, and we can verify that.
Right, is every epitope uncovered by every method of denaturing?
We don't know that. We don't know that.
How do you denature your cells, and how do you know it's the correct way of denaturing?
Yeah, I mean, what's the correct way of denaturation? We are using different denaturation agents, urea, GTC. Yeah.
So you have to try out different kinds of denaturing in order to...
We actually, interestingly, we get always to this, for those antibodies we tested that are anti-cryptic, we always get the same result, irrespective of the denaturation method. Obviously, your denaturation strength has to be optimal. If it's suboptimal, it doesn't work.
Question. The libraries that you are screening, where do you source them? Are these commercially available?
Commercial. That were the proof of concept that I mentioned, that were commercially available antibodies. Yeah. Yeah.
Okay, last quick question.
If you have a combination therapies with one target, so if you have kind of inhibitors, would you then test your antibodies for any of the kind of inhibitors because you'd expect a conformational change? Or would you check that the conjugation is still accessible?
Yeah. Yeah.
For example, if you look at anti-91 from AstraZeneca, then you get a conformational change.
Absolutely. Yeah. Yeah. Correct. Completely correct.